Greetings, gentlemen. So today we're going to be looking at the effects of selection on population genetics. So we're going to see how natural selection can actually change the genetics of the population itself. And there's three main ways they can do that. And then we're going to look a little bit more at that Hardy-Weinberg principle. So really looking at the population on the genetic scale. So effects of selection on population genetics. So we're going to be able to determine what's going to be happening to our population if a certain selective pressure is there. So let's go ahead and go back to our goldfish experiment. Okay, so here we have our population of goldfish, and I want to get one allele to be selected for and one allele to be selected against. So I'm just going to choose for my genotype relative fitness. I'm going to take my big R, and I'm going to bring that down to, let's say, 0.3. Let's go ahead and see how that would affect our population here. Okay. So as we can see, the big R is down, and as we're going through our generations, we simply keep losing it. And our little r is definitely climbing there. Okay, so that should give you a pretty good idea of what is happening there whenever I am selecting against one of the alleles. So if I go back to my graph here, so if this is the trait normally where we have some that have the one end of a trait, so this could be the white fish, and then some organisms have the other end of the trait, so this would be the orange fish, and we selected against the orange, so let's put the orange out here and the white out here. And so what happened whenever we allowed natural selection to kind of do its thing is that we got a shift of our graph. So our graph shifted away from the orange because they were being selected against. And we shift more toward the white. So the white increased in its number of individuals that had the white coloration, and we shifted away from the ones that were orange. So let's see what happens if we change it the other way. So it shifted in one direction, so we call this directional. So with directional selection, you are going either toward one extreme or the other. Now we just happen to choose that the white extreme would be the one selected for and the orange would be selected against, but it could have gone the other way as well. So in directional selection, the trait shifts toward one extreme. Okay, so now let's go ahead and see what would happen if we selected for something else. All right, so we're going to reset our population here. And this time, instead of our big R, so let's set that back at 1, our heterozygote is going to be selected against. So let's do that. So we'll put that at 0.3, and let's see how that affects our population. So we can see that our big R, little r is going down. Big R, and little r are going up. Okay, so we can clearly see that in our graph at this point. So let's go ahead and talk about that one. So in this one, again, in this one, we have our trait. where again we have few that are pure white, few that are pure orange, and most are in that middle, in that heterozygous region. And so this time though, we selected against the middle. So anybody that was that heterozygote, they were being selected against, and anybody who was homozygous, they were preferred. So they were able to live, they were able to pass on their genetic information. So what ended up happening is that we had more white, a drop in the middle, and then more on the orange side. So we actually get two peaks in this type. So we have a peak here and we have a peak over here, and we are selecting against the middle. So this is what we call diversifying selection. So again, in diversifying selection, it is the extremes that are being selected for, and it's the middle of the road that's being selected against. Okay. So this one is actually the most common that you'll see before a speciation event. As you can see here, if these two continue to be kind of on their own, so we have the white side and then we have the orange side, and maybe something happens to where they actually don't want to interbreed with each other, so like only orange want to breed with orange and only white ones want to breed with white ones, that will further separate, that isolation will further separate this one population and then eventually into two distinct populations. So this is how you can actually get a new species thanks to natural selection. So we got one more for you. Okay, so let's go ahead and reset our goldfish population here. Okay, so in this one, we are going to be selecting against both of our extremes, and so it's actually the heterozygote that is going to have the most benefit. So in this one, I'm going to go ahead and set homozygous dominant down to say 0.5, and I'm going to set homozygous recessive down to 0.5. And let's go ahead and see what happens to our population here. So again, nice normalized curve to begin with. As you can see, our heterozygote is starting to increase, and both of our homozygous are decreasing. Okay, so we clearly can see that there. Okay, so this time we were selecting against both the orange and the white, so they're being selected against for some reason, and it was this middle that was being selected for. So this is some sort of heterozygote advantage. And this is what we call stabilizing selection. So again, in stabilizing selection, both of the extremes are being selected against. So it's most often to find this under stable conditions. So if the environment hasn't really changed all that much in a really long time, the whole population tends to stabilize toward this middle. So extremes simply aren't necessary. We don't need that amount of variation in order to survive. Things have been, again, very stable in the environment. So we see that middle of the road happening. Okay. So now that we have those, let's take a look a little bit deeper back to our Hardy-Weinberg here. Okay, so with our Hardy-Weinberg, we really have two equations that are essential. We have the allele frequency, which is P plus Q equals 1, where P is the dominant allele, Q is the recessive allele, so therefore the frequency of the dominant plus the frequency of the recessive should equal 100%. Then we also have the genotype. So this is where our homozygous dominant, which is P squared, plus 2PQ, which is the heterozygote, plus Q squared, should equal 1, should equal 100%. So those are the only three genotypes that are possible. So if you add them up, that should equal 100%. Now this is a little bit misleading the way that they are written. We're actually just going to take pieces of each of those equations and substitute in for them. Okay, so let's go ahead and take a look at your handout here and let's see if we can figure some things out. So it says we have two alleles, brown B and white B. Of course, capital B and lowercase b, so our dominant and our recessive. Oops. Exists for mouse coat color. In a population of mice whose gene pool consists of 500 alleles, 301 of these alleles are brown. Calculate the allele frequencies. So to find the allele frequency, you take what you want and divide it by the total. So frequency is what you want, so whatever the question is asking for, divided by the total amount. So in this case, what we want is the brown allele frequency. So it says we have 301 brown alleles over a total of 500 alleles. So 301 divided by 500 gives us 0.60 or 60%. Okay, so if our brown allele, which of course is B, so big B equals 60%. Now it says the white allele. Now it didn't tell us how many white mice that we have in this problem, but we can certainly figure it out. And the easiest way to figure it out is using our equation for alleles. So we know that P plus Q equals 1. So our frequency, as we stated earlier, has to equal 100%. So we already know that the P allele is 60%. So plus Q equals 100%. So of course our Q is 40% or 0.40 is what we want there. So that's going to be our Q allele, or in this case, the white allele. Okay. Let's take a look at example 3. So in example 3, we're looking at a centaur population. So we have a curly tail is capital T. And straight tails are lowercase t. All right. So we have a population of 1,500 centaurs, so that's going to be our total population. We have 315 that have curly tails and 820 that have wavy tails. All right, so how many total alleles are in this gene pool? So we don't really need too much of a calculation here. If we have 1,500 centaurs and each centaur gets one genetic information from their mom, one piece of genetic information from their dad, so they have two sets of genetic information, just like all of us. So 1,500 times 2 gives me 3,000 alleles is what we're working with. Okay, so now we have to determine the allele frequencies of capital T and lowercase t. Okay, so in this case, I know, and this is one way to go about it, is that I know that 315 have curly tails. So 315 are curly. And so those are individuals, which means that in terms of their alleles, each one of those has two alleles. So they have two capital T's, right? So capital T, capital T. So 315 times 2 gives us, but then remember that we also have the heterozygote that we have to be thinking about. So the heterozygote is wavy. So in a heterozygote, we have a capital T and a lowercase t. So in the case where we have, how many does it say, 820 wavy tails, we have 820 wavy tails. That means that we have 820 big T's and we have 820 little t's. So since we're looking for the big T population, we're going to add that 820 to it here. So we have 0, 5, 1450. So we have 1450 our alleles, I should say, that are big T, out of a possible 3,000. And so that is going to give us our 0.48 or 48%. Now again, the easiest way to find little t, rather than going through all of that just for the other side of this, is again, I know that my P plus Q equals 1. I just found that my big T, or my P, is 0.48 plus Q equals 1. So therefore my Q must be equal to 0.52. All right. So hopefully this is starting to make some sense. Okay, so it says in humans, attached earlobes are caused by the inheritance of two recessive alleles. So an attached earlobe is recessive. And free earlobes are the result of inheriting at least one dominant allele. So we don't know if they are homozygous or heterozygous if we're looking at these free earlobes. And the frequency of the recessive allele is 0.7. What would be the frequencies of all the rest, assuming, of course, Hardy-Weinberg equilibrium and all the assumptions that come with that? Okay, so it tells us the frequency of the recessive allele is 70. So what is my recessive allele? My recessive allele is little r. So I already know at this point, little r, or q, is 0.70. So let's go ahead and put that in. Again, that just came directly from the question itself. So what am I going to find next based on that information? Well, it's really easy at this point to find your p. So p plus q equals 1. So therefore, my p plus 0.70 equals 1. So p must equal 0.30. Awesome. So let's go ahead and put that in there. Great. Okay, so now my genotypes. Oh gosh, they didn't really tell me too much about my genotypes, but because I know my allele frequencies, I can figure it out. So I know, for example, that Q is 0.70. So based on this information, I should be able to say that I know my Q squared. I can figure out Q squared, which is the frequency of my homozygous recessive, must be 0.49. So all I have to do is take my 0.70 and square it, and I get my 0.49. So I'm going to go ahead and put that up there. So my homozygous recessive, so that's little r, is 0.49. Now, I can do that exact same thing with my p. I already found out that p is 0.3. So p squared is 0.3 squared. So 0.3 squared, now be careful with this. 0.3 squared is 0.09, 9%. And so that is the frequency of my homozygous dominant. Okay, I'm just going to put that up here. 0.09, great. Okay, so now how do I figure out the heterozygous? Well, I know that my genotypes, I can only have homozygous dominant, homozygous recessive, and heterozygous. So if I add them all up, they should equal 1. So p squared plus 2pq plus q squared equals 1. So again, homozygous dominant, homozygous recessive, and there's the heterozygous. So my p squared I found to be 0.09. My 2pq, that's what I'm trying to find. And my q squared was 0.49 equals 1. Okay, so if I solve for that, that gives me my 0.42. So just a quick thing too, I didn't have to solve it this way. I could have just taken this part of my equation and found it. So I know that 2pq is my heterozygous. So 2 times my p, which is 0.3, parentheses better, times my 0.70 gives me the exact same thing. So it really doesn't matter which way you choose to go about it, you're going to get the exact same answer. So 0.42 is what we get. Okay. So let's go ahead and look at number 3. All right. There we go. Suppose the allele frequencies for the autosomal gene for eye color in females are B equals 0.4 and little b equals 0.6, where B is brown, big B is brown, and little b is blue. Okay, so let's assume these Hardy-Weinberg conditions. Here we go. So what are the genotypes of the population? So let's write this down, what they gave us. So they gave us that big B, brown is, big B is 0.4. And so little b is 0.6. Awesome. Okay, and so now I need to find my genotypes. Let me write here to the side again. Big B is P, little b is Q, my dominant and my recessive. So P squared is 0.4 squared, which is 0.16. Q squared equals 0.6 squared, so that is 0.36. Okay, so then 2PQ, so 2 times 0.4 times 0.6 is 0.48. Okay, so now I can put those values in here. So again, big B, that is my homozygous dominant is 0.16. Big B, little b is 0.48, my heterozygote, and little b is 0.36. Now it says, what are the genotypes of the 5th generation? Well, that's not going to change. If we're in Hardy-Weinberg equilibrium, our genetics do not change for our population, so they're the same. So kind of a trick question, I guess. Unless, of course, you know your Hardy-Weinberg. All right. And then the phenotypes of the population. So here you got to be careful. It said in the question that for brown, we don't know if they are homozygous or heterozygous. All they need is one dominant allele in order to show the brown trait. So where my blue is nice and easy, right? That is going to be 0.36, but my brown is both Q squared and 2PQ. So my Q squared again was 0.36. My 2PQ was 0.48. So that's going to give me my 0.64. You could have found it, I guess, at subtracting 100%. Again, there's several ways that you can go about this. So let's do one last one together, and then you guys can be on your own for some practice. So number 5 here, if 18 out of 50 lizards sampled has the recessive phenotype for short tails, little t, what would be the proportion of T in the population? So let's go ahead and do that. So it's telling me that little t, okay, is 18 out of 50. Okay, so little t, what part of that? Well, that's a genotype. Okay, I've got 2 alleles, so that's a genotype. So that must be my Q squared. So whenever I take 18 divided by 50, that gives me 0.36. So I know that my Q squared is 0.36. Great. Okay, and it's asking me in A, what is the proportion of my T allele? So not my genotype, but of the allele itself. So how can I find my Q if I know Q squared? Well, that's pretty easy. All I have to do is take the square root of Q squared, and that will give me Q. So if I take 0.36 and I take the square root of that, I know that my Q is 0.6. All right, great. So then what would be the allele frequency for my dominant allele? Okay, so my dominant allele, which is P, I know that P plus Q equals 1, and I just solved for Q, didn't I? So P plus 0.6 equals 1, solving for P, I get P equals 0.40. Great. Then it says, what percent of the population would be heterozygous? So isn't this nice? I don't even have to solve for my homozygous dominant because the question isn't asking for it. So all I have to do is take 2 times P times Q to find my heterozygous. So 2 times my P, which I found to be 0.4, times my Q, which I found to be 0.6, that's going to give me 0.48. All right. So go ahead and practice on your own. The next few have the answers given to you. And then, of course, go to your GIM kit and be practicing there as well. If you have any questions, and this is tough stuff, guys, this is tough. If you have any questions, make sure that you are coming to office hours so that you get this figured out before the test. Bye, guys.